will be a webinar on the identification features of the family Fabaceae and application to selected species. So this webinar is given for the analyst short course at the Iowa State University, and I want to thank them for inviting me to give this webinar. It's always a pleasure of mine, and I really hope that you'll enjoy just this little overview of this family. So as you can see up there, my name is Jennifer Neudorf, and I am a technician with the Canadian National Seed Herbarium in the Seed Science and Technology section in the Canadian Food Inspection Agency up here in Saskatoon. All right, so our objectives for this webinar will be number one, very simple, to become familiar with some of the structures and features of the seeds of the Fabaceae family. And we'll be looking at one subfamily in particular, and just some tips and tricks that analysts often use to gather information about their identity, and then how to apply this knowledge we'll be going over some fruits as well. So it'll be botany, it'll be both seeds and fruits, and to distinguish some selected species. So I have a couple of species that I'll be looking at to illustrate some of the points that I make today. So taking a look at the Fabaceae family, and I have some, if you can see my mouse here, I have some pictures of some of important, just to illustrate some of the important species. Here on the left-hand side of the screen, we have some species that are of agricultural importance. And then on the right-hand side of the screen, we have one species in particular of invasive importance. So the Fabaceae family has just over 600 genera and about 18,000 species. That's a lot. Now some of you may know the name Fabaceae. That may be new to you, and you may have heard the scientific name for the family as Leguminosae. And that's still, it's considered an older name, and it is conserved by the scientific, nomenclature committee, even though it doesn't follow a conventional practice. Now, the Leguminosae family is named that because of the unique fruit called the legume of the family, and I'll show a close-up and talk about that fruit a few slides in. Now, the agricultural importance of this family is paramount, and it is used, sometimes the seeds are used, as we see down here in the soybean, and also in the peanuts, which I always thought was very interesting. If peanuts are underground and are still a legume, how do they get there? So hopefully this picture can illustrate how the peanuts are from legumes that are pushed into the soil as they mature. It's really cool. And then we also have importance to use as forage or as honey production, as we see up in the alfalfa field in the upper part of my slide. And another thing it's used for, due to the nitrogen-fixing properties of many members of this family, where they fix atmospheric nitrogen using a bacterium near the roots, and they use that, they convert it into a usable form. So they're also called, there's some species that are called a green manure. So it's a way of enriching the soil by having these, what's called a pulse crop or a green forage crop on top, and then working it into the soil. Now, over at the other side, we have some of the invasive species, and there's one in particular. So I'm talking about kudzu here. So you can see below the immense biomass that kudzu puts out is covering this house down in the lower right-hand corner. And up here is an example of the flowers of kudzu, which I must admit I haven't seen before. They are beautiful. And apparently they smell like a combination of Concord grape and strawberries. I've never been close enough to find this out, but some of the southern states often use the flowers to make a very beautifully scented jelly or use them for candle scents. It's kind of interesting. Another member of the Fabaceae, which can be quite invasive, are acacia trees, and we'll look at the seeds in just a minute. Oh, and also mesquite and gorse, or Scotch broom, are also members of the Fabaceae. Now, let's look at the next. As I said, we'll take a look at the family where I talked about the invasive species Acacia, and it comes from, subfamily. There are 3 subfamilies in Fabaceae that I'll mention, and we'll only cover one in particular, and that is the subfamily where all of these pictures are from. So backing up a bit, we'll look at that Acacia subfamily, which is the Mimoisidae. So anything you hear of mimosa trees, those are all belonging to this subfamily. And they have very distinct flowers, and they're more found down in the south. South and tropical. So these flowers here, look at all the stamens just sticking out like crazy. It's just this big puffball of a flower. And the seeds are also quite unique. Now, you haven't seen too many Fabaceae seeds yet, but you will definitely see how different, the subfamily we'll be looking at, how those seeds are quite different from this Mimoisidae seed. So the seeds of Mimoisidae are recognizable in that they're flat. You can't see that here, but they're flattened. And specifically, they have a U-shaped structure called a pleurogram. And this structure is just an area of epidermal or seed coat cells that are a little bit different and crack easily to allow water to come in. So it's like almost like a water gate or a sluice gate, just kind of controlling when the water comes into the seed for germination. And then the hilum of the seed is just tiny, tiny and up top. Now, folks may have run into this, the seeds of the subfamily more than they realize. I'm not sure if it's around anymore, but there used to be a type of seed jewelry, and the seeds were called apple seeds sometimes, so they aren't actually apple seeds. Unfortunately, I don't have a picture of that, or lead tree seeds. And these are seeds of a genera, or a genus, I should say, belonging to the Mimoisidae called Lucana. So if you do have any old seed jewelry, it was more popular in the 60s and 70s perhaps, or apple seed jewelry, take a look under the microscope, bring it into the lab and look at it under the microscope, and you will see these very flat seeds and you will see the pleurograms. So it would be really cool if any of you have that. Now, the next of the 3 subfamilies we'll just be touching on is the Casalbinoideae. I don't run into this subfamily this much, as you can tell. This is another tropical subfamily, and the seeds, again, and the flowers are different than the Mimoisidae. So this subfamily flowers have those 5 petals we're used to seeing rather than that spray of stamens. And the stamens, you can see the anthers, the pollen producing tips of them are quite distinctive. They do have a legume, of course, and the seeds inside have that pleurogram, very similar to the other subfamily, that area of different cells in the seed coat that can control water coming in, or it's the area where water can come into this normally hard seed coat of this family. And again, the hilum is just a tiny little area at the tip of the seed. Now you see, unlike the Mimoisidae, the seeds of the Casalbinoideae have they're more 3D and they're more angular or polygonal, and that's very, very common throughout the family, just a bit of a thicker seed. And this family, you may know, I think, there's a type of natural laxative called senna or cassia, if you folks have ever heard of that. It's ground up seed and it's from this subfamily. But up here in Canada, we definitely don't see a lot of this. But we do see a lot of our last subfamily that we will definitely be spending the rest of the webinar on, and it's most important for our commodities and our pulses, which is the Fabaceae. And here we have a very distinctive flowers again, and this also has 5 petals, but they're arranged in a very specific way. And that's for guiding in pollen collectors right into the center of the seed. So you have your one big petal up top here, and that's called the banner. And you can see some veins or some stripes that act kind of like arrows pointing the bee or the insect in towards the middle. And then you can see 2 other petals that protect the legume in the middle, and then underneath that are other petals. So there should be should be 5 petals in all, and there's 2 that you can't see here. So we've got the one big one on top, and then we have 4 beneath, and a couple of them are hidden on this particular one. And then we have, again, the seeds that are different than the other 2 subfamilies. And these seeds are your typical bean seeds in particular, as you can see here. So they are bean-shaped, they're kind of biconvex or lentil-shaped in cross-section, 2 convex sides to them, and they don't have a pleurogram. That's a huge thing. Instead, they have other water controlling structures or different cells in the seed coat that are actually really important identification features or accessory identification features that I will be covering in depth later as well. All right, so we're going to take a look at, I'm going to start to look a lot closer at those seeds. That's what we're here for, but I'd like to back up a bit and show you folks the unique fruit of the Fabaceae, as well as how perhaps some of these seeds get these very interesting markings and identification features, how they arise in the fruit. So we have here the legume. That's a typical fruit of the Fabaceae, and its technical definition is that it's a fruit that usually splits along 2 sides. Now, there's variations on the legume where some of them are single-seeded and don't split apart at maturity, and sweet clover is an example of that. Or it is a legume, a long one, and it splits into single-seeded portions. It splits between each seed and is shed in that way rather than splitting lengthwise. And an acacia legume is an example of that, and there's a couple of examples. Sweet broom is one I'm thinking of that we have here as a native species up here in the prairies. So looking at this legume, I'd like to show you some of the remnant flower parts just to situate you. And here is that old stem, and here is where most of the flower is left behind. So you see some of the calyx, you can see a tiny little white petal, and then at the other end of the legume is a little bit of just a tag here that is a remnant, what it's called the style, and that's the portion of the ovary that supports the pollen collecting organ called a stigma, and it's just a little tag that's left behind at the edge of this legume fruit. Now inside, you can see the seeds, and you can see they're connected by a little stalk to the main nutrient vessels in that feed the developing ovules or the developing seeds. And this is called the furunculus. You can see it down here. Now when this furunculus, when the seed is mature and it falls off, the furunculus dries up, and what is left behind on the seed, that's the hilum. Absolutely. So when the furunculus drops off, a little bit of tissue is left behind as the hilum. And there are some also very interesting identification features associated with the hilum that is part of a modified furunculus. So that's pretty cool. So let's take a look. So we know where our seeds are situated. We know how they get some of those markings. Let's take a look at the seed and underneath the seed so we can see why seeds are shaped the way they are and why they have the interesting features around the hilum that they do. So now, just take a look at the split seed on the left side, and you can see taking up most of the room here is a part of the seed that gives mostly that characteristic biconvex or lentil shape to the seed. And this is 2 huge cotyledons, or they're also called seed leaves down here. So these organs contain like vast majority of the nutrition that it takes to feed a developing sprout. And you can see here the true leaves that take over photosynthesis once the sprout is established, and they're called, right now they're called the pumule. And if you were to split apart a peanut, very often you will see at least on one side you're actually splitting apart cotyledons once you take them out of the legume, and then you will be able to see inside a tiny embryo and a tiny little pumule. So I invite you to do that. It's just a really neat way to put all the drawings and all of my words into context. Just check it out for yourself. All right, so right here, so we've got the main two of these cotyledons, and you'll notice like when you split that peanut, they're flat on the inside, they squish together, and then they're rounded on the outside. So you get the main shape of our pea seed, but another part of the inner part, the internal parts of the seed that give rise to the shape is specifically the end of the embryo called the radicle. And this little lobe can stick out and give an interesting knob or a kind of a beak look to the pea seed, or it can be tucked into the cotyledons, and then you won't see it stick out at all. And then, of course, right here we have the hilum with some spongy tissue underneath. All right, and you can see that again here. So here's a non-dissected seed with the seed coat taken off, and you can see this radical lobe. It's not tucked into the cotyledons, it sticks out, but it's pretty well squished under there. And you can see the two big cotyledons, and you can see this ridge or this furrow that lies beneath the hilum, where the water, once it gets through the hilum, can be funneled, and so that radicle can soak up that water and the seed can germinate. So you can see the hilum, as I just talked about, that's the obvious feature of the seed. I mean, usually we don't peel our seeds in order to identify them, but I just wanted you to show you the internal anatomy, which gives rise to the shape that's underneath that really tough, leathery, or very hard seed coat. And you can see that obvious hilum there. And it's got, well, let's see on the next one. It has got, before we get into the specifics of identification features around the hilum, let me show you how the radicle plus the cotyledons inform the shape of the seed. So hopefully on these two seeds, we've got an alphaceae clover seed and an alfalfa seed, that you can see how those large cotyledons and the protruding radicle can give shape to the seed. So now I talked about how the radicle could poke out and give this little knob or a beak shape to the seed, and you see on this alfalfa that it is right beneath the hilum and it just butts out a little bit here. However, on many of these seeds, you can trace back and you can see the line that separates the radicle, that embryo end, from the rest of the seed by it could be a furrow, it could be a depression, so I'm calling it a radical furrow. And in some seeds like sweet clover, it can just be a line or like a smiley line, and that can be used in identification or to recognize a genus. Now here on our alphaceae clover, it's upside down from the alfalfa, so that radical lobe you can see up top, see the end of the embryo there, gives that kind of a ridge or a hump, and then you can see the border between that and the cotyledons, and you can see that as a furrow. It's not quite as, doesn't have that light line in particular. All right, now to sum it up, the features that help us to recognize that the seed we have in hand belongs to the Fabaceae are a tough and a smooth seed coat, the visible hilum, it's visible, it can be big or small as you'll see, and it's situated along the narrow edge of a seed. So the seed is kind of compressed, squished in on the sides, and that narrow edge is where all of the interesting stuff goes on, where the embryo sits. And also it's the interior, you see that two-parted hilum. And then we also have, as I mentioned earlier, there are parts associated with the Fabaceae hilum, and which I'll go into in the next few slides. But let's just, before I do, okay, I know I'm teasing. Before we do, I have some slides before that that just show you some of the diversity of the PC shape and the color and the pattern of the seed coat, okay? But while I'm showing you those pictures, I'd like you just to keep an eye out for how where the cotyledons are and where the radical lobe is, and how that can start changing the shape of the seed, as we have here. All right, so what we have is we have some seeds, and you'll notice that I didn't label them on purpose. So as I talk about them, I'd like you to try to see if you can recall what these are. They should be, at least some of them should be fairly common, and you may not have seen these if you've done some practice with analyzing, you may have seen them. Or if you're absolutely new, you may have seen these in your everyday life. You never know. So now the slide I have here is by no means exhaustive of the shapes, but it gives you some idea of the diversity of shape. Now, I'd like you to find, okay, well, absolutely, if you can figure out the names of them, that's great. But as I mentioned earlier, I would like you to try and find the cotyledons, where they are, where the radical is, and where the hilum is on each of these, and also give you an idea about the diversity of shape of the cotyledons, the radical, and the hilum, and how all three of those inform kind of the overall look of the seed. All right. So I'll just take a couple minutes here, or a minute or two, so you can think about this or discuss amongst yourselves, or just mark down on a piece of paper what you think, where you think the cotyledons are. And of course they're going to be paired, so you can just say where one cotyledon is if you want. And just take a look and see if you can see where the hilum is. And remember, it might be covered, might be rimmed or covered by tissue. And I did promise I'll go over that in detail after we take a look at the color and shape. All right, hope you have an idea. It should be fairly easy. It should be fairly easy to pick out. Let's give a few more seconds. All right, 3, 2, and 1, and here we go. Here are the answers. So find the radical, cotyledons, and hilum, and guess at the name. Hopefully you got some of the names right. If not, that's fine. You will absolutely encounter these during your work, I'm sure, most if not all of them. So up at the upper left, we have our peas. And you can see the cotyledons, or one of them anyway, and then you can see the radical lobe, but it's not really sticking out. They tend to show kind of their parts through lines, not lobing or furrowing, but the hilum's nice and big, this big oval shape. And you'll notice there's a line in the middle of that. I will talk about that in a moment when I zero in on the hilum. And then over here we have alfalfa. And alfalfa has quite a big cotyledon, kind of long, while the pea has kind of a half circle of one. One in alfalfa is long. And you can see that's the hilum. Here it is right here. It's very small and round, smaller than the pea. In this case, as we saw earlier, the radical tip does protrude as a lobe. It's really good on this seed here. And definitely notice that the shapes of the alfalfa don't seem to be consistent, and this is indeed a thing. There is quite a range of variation in the shape. Pardon me, some people have described it as almost like puzzle pieces, although they don't really fit together, but they do. They do in the pod. So that's the reason why these are such crazy shapes, is because there are many seeds crammed into a pod that's coiled. So all of those curves in the pod gives rise to kind of odd-looking shapes in alfalfa once they mature. Now for the red clover, in this case, the big cotyledon here and the smaller radical lobe diverge into a little bit of a mitten shape. And in that notch, you can see over here on the left, is the hilum, and just like alfalfa, it's round, quite small and round. And then looking over into the mung bean, we have again a very even, symmetrical cotyledon, just two kind of oblong cotyledons, and it's flat on the bottom and the top, probably from being, that's how it's squished in the pod. And we have our hilum is quite obvious. It's big and white and oval, and it's covered by a bit of tissue. And then the radical, it is a lobe. You can kind of see it over here on the left. It's just a little bit of a bump out, and it forms like more of a crest than kind of how big you have it over here in the clovers, just a tiny little crest. Good, good, good. All right, let's take a look at the color and the pattern. So again, this is by no means exhaustive. It exhaustive, but that tough seed coat has some gorgeous colors and patterns in the Fabaceae. So you can use color patterns. It's not used as often, but sometimes some particular seeds, even within a genus, species within a genus, can have just variations in color pattern, and especially in our cultivated species, like up here in our lentils. Different varieties can have different colors, even though they're all the same species. So we use more the shape and then the hilum and associated parts that allows us to recognize genera. Now down here, I just have to point this guy out, the blue lupin seeds, really interesting. This particular one has a marking, which is basically an arrow that goes through that hilum that points to where a certain area or a hilum accessory part that's called the lens. And it just, this helpful arrow pointing right to the lens, it's just so lovely. But it's giving us a hint that maybe I had better show you folks what I'm talking about, just some microscopic features of the hilum. Oh no, before I do that, I'm teasing so much. Before we do that, we're going to take a look at some of the changes that can occur with the pea seeds, seeds in the Fabaceae. So within this family, it might have some unique properties to the seed that might throw a little bit of a twist into identification. And it kind of makes the reason why we don't really hang on too hard to the color to help us to tell what species we're looking at. We definitely look a bit more at pattern, but color can change. Many species, they start out as kind of greenish or yellowish, and then that color darkens as they age. So older seeds, and these are seeds that maybe, you're not necessarily going to get super old seeds, of course, in the seed that you analyze, right? It's going to be new and fresh. But when you have a reference collection that's been there for a while, when you have specimens that you're comparing to on your slide, those are going to darken with age, and it may throw you off. So I just want to give you folks a heads up. So this is what happens when a seed gets old. It doesn't gray up like mammals do, but they darken and turn brownish. And also appears like this green pigment is what breaks down over age. I'm not too sure, but they turn caramel, camel-colored, and brown. Now another thing that happens, now I'm not sure if that would also signify that they're losing viability, but it does signify that they're older seed, not so fresh, so I imagine it would. Now another thing that can happen, in my experience, seeds of Fabaceae, especially those older seeds from crop species such as clovers or vetches, seem to be commonly infected with a fungus that appears as silvery threads or a powdery bloom on the seed surface. So this isn't a seed feature, and folks that maybe haven't been experienced with the seed in hand may see this and see it as a color pattern, but it's actually not. It's just a kind of a normal fungal infection, and you're not going to catch anything, but it may confuse you a bit. So just to let you know about that. All right, without further ado, let's get looking close up at that hilum. Now we're getting finer and finer features, and what we look at to try and make sure to try and identify the genus, to distinguish between genera, we definitely start looking at the hilum. So remember I mentioned that there was one of the hilia were two-parted, and that's a feature of the subfamily. And you can see the seam in the middle. So this is the seam that corresponds to the border between the cotyledons, and I suspect there might be a bit of a trough beneath that. So that's what you're looking at. You're looking at the border between the cotyledons. I'm not saying you're looking into the interior of the seed per se, but this corresponds to that border. And it's very, very common within this subfamily to have that two-parted hilum. Now, the location and the shape of the hilum tends to be conserved between genera, and it's a really helpful feature. Now, depending on how it develops, the position of the hilum may be different. So some seeds, such as here in the gorse, the hilum looks like it's on top of the seed. And then many of the species that we have, the clovers and sweet clovers, the hilum looks subapical or near the top. And on many of our bean species, the hilum is almost in the middle of that narrow edge along the side, or technical speak is that it's lateral. So position of the hilum can be different between genera and help you to recognize what genus you're looking at. Another thing about the hilum that can also help, as you saw, diversity in the hilum shape and size. So I have, I'd say 3 different basic shapes, but it's almost like 2 times, and one's a little bit longer, but let me go through these in order. So we have a round hilum found in many clovers and many forage species. And you'll notice there's a ring of tissue around this hilum. That'll come in later. Or we can have an oval hilum. And in this case, I said there was a short oval hilum, which is about 2 to 3 times as long as wide. And in this case, in the mung bean, it's covered by tissue. And then we can have a long oval hilum, an oval hilum that is several times longer than wide. And here in the lathyrus, in the sweet pea or flat pea, we have quite a long hilum. And again, you can see some tissue associated with that. Okay, so you could say there's 2. It's round or oval, but I made a few finer, broke one of them up into 2 shapes because one's quite consistently longer than the other. Okay, so I mentioned about the tissue associated with the hilum. And I don't know if you remember, when I was talking about the fruit, I also mentioned that the furunculus, that stalk that connects the developing seed to the nutritive tissue in the ovary, or what's going to become the fruit or legume of the Fabaceae. It sometimes comes off, no problem, and sometimes part of it remains behind in a piece of tissue called an aril. So they may be ring-shaped. You might have a ring-shaped aril, as you saw on the previous slide, and on this Lespedeza, and quite simple, so that can be an important feature. You can have a lobed one, like up here in the gorse. So if you were to look at this top down, you would see it's actually 2 lobes, this piece of tissue. And then you can have a long one, like on this hairy vetch. However, it's usually broken off once the seeds are mature. But if you can see a picture of it, it actually can extend almost the length of the hilum. The hilum's just underneath it there. It's kind of a hook. And then here you have a bit of a raising of the hilum. It can just cover the hilum, as you saw in the mung bean. And here in a different species of the same genus, Vigna, as the mung bean, you can see it here. So it seems to be a common thing of the genus Vigna, which the mung beans and snap peas and cowpeas are found in, as well as adzuki beans, that the furunculus almost partially breaks off, leaving some spongy tissue behind and obscuring the hilum. As an aside, remember I mentioned earlier about the problems with older seeds, and I guess you can see this guy's graying up here. There's a bit of that powdery mildew I talked about, which is just a consequence of an older seed. Now, why is it here? Well, it definitely aids in identification, but it serves a purpose. It serves a dispersal purpose. So it's used to entice animals or insects to disperse the seed. So what they do is they transport the seed, drag it away from the plant and chew off the hilum or eat the hilum. So it's a bit of a tissue, a nutritive tissue reward. And what happens to the seed is that it's dispersed away from the parent. So it's pretty cool. And also there's an example, our good lupins here again, where the seed coat seems to form a dam almost around the hilum, but that's not a aril, pardon me. This arises from the seed coat, not from a modified furunculus. And it is the same consistency as the seed coat, and it's not a food reward for dispersers at all. It might, in some cases actually, to be honest, I suspect, I don't know too much about the relationship between dispersers and seeds, and especially I wonder with the Fabaceae, because they're such large and heavy seed, I can't imagine an ant dragging it around on the ground, and I'd hate to meet the ant that can drag one of these around, to be honest. And I also wonder if the aril forms a way of holding water close to the hilum, because, you know, in many of these temperate species, in the summer we get our rain showers as quick events. So perhaps the plant or the seed takes advantage of that by absorbing water using that spongy tissue to keep, like a sponge, to keep water close to the hilum to ensure a water supply as it germinates. So again, those are my ideas, but I don't know for sure and I haven't read up, so definitely fact check me on that. Okay, now taking a close-up look at the hilum. I mentioned that there were some other parts. Now, the hard seed coat in the Fabaceae really protects it from water, well, it protects it from predators, from seed eaters. And water, however, can't really get into the seed very easily to break that dormancy. So you've got your hilum here and it's covered with tissue, as I mentioned, may act as a sponge in some genera or species. But you can see to one end of the hilum close to where the radical lobe is. So if you had the seed in hand, you could probably see this bit of a lobe sticking out. So that's where the radical lies. Between the radical and the hilum, there's a tiny hole into the seed, into the inside, and we call it a micropyle. And this really isn't used specifically in identifying what genus you're looking at. However, it can help if it is visible. Many times it kind of seems to blend in with the hilum, but if it is visible, it can definitely be used to orient your view to try and find the other end or the other accessory part, which is called the lens. And that's a very important part to help identify what genus you're looking at, what shape it is, if it's there, what color it is, how far it is away from the hilum. All of those are identification features that can be used to distinguish genera and in some cases species. So I picked a particular one in this garden bean where the lens is very prominent and very interestingly shaped. And in the next few slides, I will show you some other example of lens where you can see that the shape can vary. And hence, since it can vary, it can be used as an identification feature. And as I mentioned, there is a lens. Well, there's a lens on every pea seed, and specifically in the subfamily we're looking at, the Fabaceae, and the lupin with that helpful arrow that points to the lens. You can see in this case the lens is quite a distance away from the hilum, while in the garden bean it's a lot closer. So to sum it up, associated with the hilum are the lens and the micropyle. And micropyle not really used in identification, lens definitely is, but the micropyle and the radical lobe can help you to orient yourself knowing that the lens is going to be on the opposite side of the hilum from the micropyle and from the radical. Now, before I move on to show you some different lenses, some different hilum accessories, I just want to let you know the reason why there's a lens. It's not just for identification, but again, as I mentioned with that tough seed coat, this is similar to that, analogous to that pleurogram, that portion in the other two subfamilies that have a bit of a weaker cell, cell walls or cells, different kind of a shape of cells that with not freezing and thawing, but drying and soaking can kind of contract and expand and form little cracks where water can get in. And you can see by the way it sticks out, I wonder also if maybe it can be abraded as well, just through natural processes. So here we go. Of course, I'm not only going to show you the picture, I'm going to ask you to do a quiz along with it. So much like quiz number one, this quiz is in two parts. For each of these three seeds, label three things. Where's the hilum? Where is the radical? And where is the lens? Now remember that the lens is going to be on the opposite side of the radical. And the part two, like part one, see if you can recognize them. You may have run across them at work, or you may have run across them just in your everyday life when you're doing some cooking. All right, so I'll give you a moment or two for that, to discuss it amongst yourselves or to write some stuff down. Where's the hilum? Where's the lens? Where is the radical? I don't know if you'll be able to see the micropyle, but it'll be really near the hilum. I'm taking a close-up look here and I can't see them, the micropyle, but definitely I can see where the radicle is and then where the lens is. All right, pretty easy. There's only 3 seeds. Okay, the answer's in 3, 2, 1. Here they are. All right, so on the left-hand side, we have a chickpea. And I believe the chickpea gets its name from having a tiny beak. And that tiny beak of the chickpea is indeed the radical lobe. And then you have the hilum right next to it, and you can see it's round, kind of in a, almost in a little bit of a depression there. And then you can see, hopefully, from back there where you folks are, you can see there's a line. It's a 2-parted hilum. Now, between, on the other side of the hilum from the radicle, we have the lens. And it's not sticking out like we saw in the garden bean. It's pretty well flush with the seed, but you can see it's a different color. And as well as I almost can see it's like a tiny depression around it, or this looks like the seed coat to quite a distance looks a little bit different. I'm not too sure if that's actually associated with the lens, but there it is. A little bit of a different color for the kind of different cells that make up the lens area as opposed to the rest of the seed coat. Over here in the lentil, we do have a radical lobe, but like in mung bean, it looks almost like more like a radical crest. And then you have the hilum, quite visible. And on the opposite side, from the radical, you have the lens. And much like chickpea, it doesn't really stick out. It's just a kind of an ovalish area of darker colored cells that are different than the other cells in the cell wall of the seed. And I think I can see that kind of that fungal kind of a bloom on the surface of that mildew bloom as well on that one. All right, here we go for our black medic, a nice fresh black medic seed. You can still see some very nice dark green pigment there. And easy enough to see the radical. That's kind of the pouty lip. Some people describe that as the black medic seed to recognize it because it looks like the profile of a pouting guy with a big old chin. And there we have that lip, and then you can see the round hilum right above it. And the lens is a little bit harder to see. It does bump out a bit, a little bit of a forehead there. But more specifically, take a look at that color. You can see it's a darker green, a big oval area. And if you can see close up the picture, if you get it as if you're able to see it close up on your screens at a later time, you can see that it almost looks cracked. You can really see a difference in those cells in the cell wall. Good. So let's take a look at that hilum, and specifically the hilum and the lens, and see if the distance from the hilum can help us to identify selected species. So here we have the vetch that can be used as forage, and there's a couple of them that are weeds. And these vetches, there are 3 species that are quite similar looking. We've got, on the left, we've got hairy vetch. And on the right, we have common vetch, which are commodities. And in the middle, we have a weed species. I'm not sure if Vicia cracca is used as a commodity or if it's truly a weed. I'm not too sure. But anyway, it's called wild vetch, so you can imagine that it's often an impurity. Now, the 2 on the ends, let's take a look at the hilum. Now, one thing you can notice right away is that the hilum length is different between our 2 bookend species here. We've got a shorter hilum in hairy vetch, and the hilum is at least 1 time or 2 times longer in the common vetch. Taking a look, you can see the lens. It's a little bit raised area, and you can see those different cells, especially on this picture with how it reflects the light a little bit differently. And it used to be that I've been able to see some differences in distance between the 2, but they haven't been consistent, unfortunately. So I can't say for sure that there is a difference in distance to distinguish these two species. It's more in the lens, or pardon me, the hilum length I find. However, in the middle, this wild vetch does have a lens that is quite a bit of a distance away from the hilum, thus making it distinguishable from our two crop vetches. So you can see quite a bit of a ways down. And then the hilum's also going to be a bit different as well, of course. And that's mostly what's used, or when I learned how to tell these vetches apart, we definitely concentrated on the hilum, the length, and maybe some tissue, maybe that seam in the middle sometimes is a lighter color in some species and is the same color as the seed coat in others. Definitely when you folks learn vetches, you'll probably get more into this. But when I took a look, I did notice that the distance between the hilum and the lens just gave me yet another kind of a check mark to put when I was identifying a species, so I'd like to pass that along to you folks as well. All right, now there's one more vetch, and again, it's another crop vetch. I don't see it as much anymore, but it is definitely on our grade table in Canada, and it is called Hungarian vetch, Vicia pannonica. This one's really cool. The lens, in this case, the lens is on the absolute opposite side of the hilum. Pardon me, it's on the opposite side of the seed from the hilum. I don't know why, it just is. Makes it easy to identify, thank goodness, because you can see the color is, I can't say too easy on these, but trust me, they're both a reddish brown mottled with black, and so is this guy. So you can see how those crop vetches look very similar, but always, 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 you got the hilum on one end of the seed and then go all the way over to the back end of the seed, and that's where the lens is. It's really cool. All right, so I'm going to end our webinar. Thank you guys so much for your patience. Again, I love my quizzes and kind of helps you to recall some facts rather. I know I could just be talking, but I want you folks to also kind of incorporate some of this knowledge and remember some of it when you do get into work. So we're going to just do a summary and take a look at some questions here. So the first question I would like answered is, what is the unique fruit of a Fabaceae called? It's characteristic to this family of plants. On the two seeds below, our L-site clover and our alfalfa, I'd like you please to find the cotyledons, find the radicle furrow, that is that border between the radicle and the cotyledons, that's away from the hilum, and please find the hilum as well as the lens on these. Now, I'm curious, when you look at these two seeds, and let's say you were to distinguish them, yeah, indeed they do look different, but I'd like you to kind of take a look and question, why are they different? What are those parts of the seed I talked about for this webinar, and what are their characteristics for each seed, and how do they contrast? So how would you be able to tell these seeds apart? And the hint of what you look at is up here. Take a look at the shape and at the size of it, of the seed. Take a look at the cotyledons, they're going to be there, but they're going to be different. They're going to show a different shape to the seed. So take a look at the shape. Take a look at the hilum. Is the hilum different? It might be different. It might be the same. If it's the same, it's not a good feature to tell these two species apart. So I'll just give you some more time to finish off this quiz. Just a few more moments. And again, you can write these down, you can mark them in your mind, you can just discuss them a bit. All right, answers are going to incoming very, very soon. 3, 2, 1, here are the answers. All right, the unique fruit of the Fabaceae is called the legume. a fruit that normally splits on two ends. Some of them are single-seeded and don't split. Some of them split crosswise, but by and far the vast majority are split along two sides, thus releasing the naked seeds onto the ground. And the cotyledon, you can see at least one of them in this picture. And then you can see that radicle, that kind of a hump coming up on the L-site clover, and one coming down along the narrow edge of the seed on the alfalfa. And you can see the furrow in this case, a radical furrows. You can see a depression, kind of how the light is here on the L-site. And on the alfalfa, not so much, it's kind of a straight-on view, but you can definitely see a lighter area in that radical furrow. The hilum is very similar on both seeds, round with a little bit of tissue around it. And the lens on L-site, you can see that reddish area below the hilum on the opposite end from the radical lobe. You can see that. That's the lens right there, just that patch of color. And then again, it's a little tougher to see it. You can see it bumping out a little bit, the lens on the alfalfa. But if you took a close-up look, you could also see that the cells look almost a little more glassy or cracked. It's really cool. So definitely, when you do have a chance, take a close-up look. Now, similar features as I mentioned. We've got a round hilum. We have a large radical lobe on both of those. But alfalfa has a longer seed, as you can see, over 2 millimeters versus 1 millimeter, a little over 1 millimeter on the L-site. It's a different shape. L-site clover is round, while alfalfa is a bit more bean-shaped. And the color of the seed is different. I mean, these things would pop up right away to you folks. So you got also the pattern's different. You got a solid color on the alfalfa, and then the L-site clover is mottled. And then on the clover, the lens stands out. It's a color nowhere else on the seed. It's that reddish color. And that's very common to have a very darker lens, and that really helps out with, again, confirming identification. I don't know, when I do identifying Fabaceae seeds, I definitely use the lens quite a bit, just as that final confirmation to what I suspect the identity of the seed is. Again, thank you so much for your attention. This is always a blast for me, and I really hope that you folks enjoyed it as well and learned a bit about the Fabaceae family, and specifically our very important subfamily here in the temperate areas of the Faboideae. So, the beautiful images that you saw today came from National Seed Herbarium specimens and were taken in-house with our imaging equipment. We're very, very proud of that. And any questions or comments, please send it to our email address at ssts@inspection.gc.ca. I should also mention at this time that the Government of Canada is now having a different email. So by the time you get this or want to send us some comments, we may have a different email address. I didn't update the slide, unfortunately. So if that email probably should forward, but just in the future know that we're going from inspection.gc.ca to @canada.ca. All right, again, thank you guys so much, and I really hope you enjoyed yourselves, and definitely good luck in the work that you do, and right on. All right, thank you.